It's now my pleasure to introduce our speaker. And Dr. Corrie Moreau is the Moser Professor of Arthropod Biosystematics and Biodiversity at Cornell University in the departments of Entomology and Ecology and Evolutionary Biology. She completed her undergraduate and master's degrees in biology at San Francisco State University, but most importantly, she earned her PhD in evolutionary biology from here, so she's one of us, and she worked at that time closely with and was advised by Professors Ed Wilson and Naomi Pierce, so I believe that must have been wonderful for you as well as for us. And she was also a Miller Fellow at the University of California in Berkeley. Before leaving for Cornell recently, she has also worked as a curator at the Field Museum in Chicago for 10 years. Her research is on the evolution and diversification of ants and their symbiotic bacteria, and she leverages molecular and genomic tools to address the origin of species and how co-evolved systems benefit both partners. She also pursues questions on the role of biogeography and symbiosis in shaping macroevolutionary processes to better understand broad-scale evolutionary patterns of life. In addition to her passion for scientific research, Dr. Moreau is also engaged with efforts to promote science communication and increase diversity in the sciences, a cause that we all support, including helping and mentoring local high school students while she was at the museum, and I believe also now she's at Cornell. And I should say that if you haven't seen her video on bullet ants that is part of the Field Museum's Brain Scoop series, Google it. It's on YouTube, and you should do it as soon as you leave this lecture theatre, because watching her milk the ant to analyzing its venom is truly amazing, and more than 2 million people agree with me because that's how many people have seen this video. Anyhow, she's also the author of many scientific publications and recipient of various honors. In 2018, she was elected to be a fellow of the American Association for the Advancement of Science, and she has been identified as a woman of impact by the National Geographic Society. In 2016, she was appointed to be a Kavli Fellow of the National Academy of Sciences, which is an honor given to only a very few distinguished young scientists. Tonight, she will share her research on the critical roles that microbes play in the biology of ants. So please join me in welcoming Corrie Moreau.
Thank you. Thanks for that very nice introduction, and it's really great to be here, both at Harvard because it's sort of my intellectual home where I did my PhD, but also to be sharing this with a community online so that people who aren't just in this room can also enjoy the programming that you guys are hosting. So I thought for the young people in the room, I would talk a little bit about my path to science. So this is me at 5 years old. I actually remember this day because it was school picture day. And I woke up and my mom had been promising to sew my butterfly patch on this jumper, and she had not. So I begged and pleaded until she did. And she kept saying, "But Corrie, the picture's from here up." And it's that one moment at 5 years old where you feel so vindicated when you get your picture back. But it does demonstrate that I was interested in bugs from a very young age. And so thinking back at myself at 5 years old, did I ever imagine that I'd sort of have the career I've had, where I've had the opportunity to travel the world collecting bugs and doing science on bugs? So I thought I'd talk a little bit about how I became a scientist. I grew up in New Orleans, Louisiana, where there's lots of bugs. So of course I kept them in jars in my room and in boxes under my bed. But really I didn't know that you could really have a career as a scientist. I fastidiously watched PBS and thought, every scientist that the world needed was already on PBS, so I didn't know that I could also be a scientist. And as I sort of, grew up and cultured both my love of insects, but also started to realize the pressures that young girls were feeling to not necessarily be engaged in the sciences, and certainly not to like bugs, I think that's sort of where my passion came from, really wanting to promote young women and people of, different cultures and communities to be engaged in the sciences. Now, luckily for me, the one thing I did know when I went away to college is that I absolutely wanted to study evolutionary biology, and I absolutely wanted to study insects. And I was very fortunate to find a home, a scientific home, almost immediately in undergraduate where I got to do hands-on research. And that sort of has catapulted me on this career where I've been able to go from, great institutions, working on science. Now, throughout my career, natural history museums have been a critical part of that. Of course, doing my PhD here at the Harvard Museum of Natural History, having worked at the Field Museum for 10 years, I did my master's thesis at the California Academy of Sciences, and I'm now the director and curator of the Cornell University Insect Collection. So really, even though a lot of what you're going to see today, the tools I use are molecular and genomic, natural history museums and the collections in them are at the core of what I do. I was also fortunate to have really amazing mentors. My dissertation committee was made up of sort of the rock stars of science. In fact, you can see some of them here today, and in fact, Scott Edwards is even here in the audience, and I got to see E.L. Wilson earlier today. In that, really, what they did is they sort of not only cultured my intellectual curiosity, but showed me that I could be a scientist and really helped me navigate the system. But now what I'll do is I'll switch gears and I'll talk about why I spend so much time studying ants. I have been asked many times in my career, why would you focus so narrowly just on ants? But it turns out that there are more ant species than all the birds and mammals added together. And that diversity isn't just in the number of species, it's also in the forms and shapes of them. You can see some of that diversity here. It's also in their behaviors, the way that they interact in the environment, what they eat, where they live, and they're distributed across the globe. So it really is a great group of organisms if you're interested in trying to understand broad questions in evolutionary biology. In addition, ants have a very interesting characteristic, as do many other Hymenoptera, the wasp, bees, and ants, and that they're a female-dominated society. So I would argue that every ant you've probably ever seen in your life is female. So females are responsible for all the work of the colony. They're the ones that are out foraging in the environment and bringing food back to the nests. They're the ones that are having those battles to the death on the cracks of your sidewalks. They're the ones that are sort of building the nest. So males and virgin queens are produced once a year when the environmental conditions are right. And that's explicitly so they can go off on a mating flight. So males have given no contribution to the care and growth of the colony. They go off on a mating flight, copulate with one or more virgin queens, and then die immediately. So they really have the sole role in the colony of reproduction, of passing on their genes. That new queen will go off and find a new place to start a nest and start laying an all-female army to sort of grow the colony up. In addition, ants are among the world's first farmers. So if you've ever been to Central America or South America, you've probably seen these very charismatic ants walking around with these parasols of leaves above their heads. And the reason that they're going into the canopy and cutting off pieces of that leaf with their mandibles is to carry it back to the nest and grind it up. But they don't actually eat the leaves. What they do is they grow a fungus, and that's the picture on the bottom, that they then eat. Their sole food is this fungus or mushroom that they've grown themselves. And so they've been doing this for over 50 million years. In addition, of course, there's this sort of charismatic group of ants, the army ants, which everyone loves and also fears. And part of the reason that people fear them is because they go off on these massive raids. But they also have these really interesting soldiers with these sickle-like jaws on the front of their heads. And that's so that they can subdue prey or they can deter predators. But people in Africa and also in South America have long known that you can use them as a medical aid. If you're ever in the forest and inadvertently get a terrible gash or cut, you can use them as sutures. And so what you'll do is you'll close the wound, let the ants bite on each side of that wound, and then just wait. And as your body naturally heals, parts of the ant will fall off, and you can see by the end, only the head capsules are even attached anymore. And once you've fully, healed, you can just pull those head capsules off. So if people ever tell you ants aren't important, you can point to this example. So why did I tell you all these fun facts about ants? Clearly, I love ants, and I think all of you should as well. But ants are important because they're found across the planet, and they play critical roles in sort of ecosystem services. They're incredibly important for aerating soil and for increasing the degradation of organic matter. They also even keep back pest species. And because ants are found across the globe, with most of their diversity in the tropical regions, so you can see on the warm parts of the map are where we have the most species of ants, That means that I've been able to travel around the world collecting ants. And I consider myself very fortunate, because if you went back to 5-year-old me and told me I would be like those people I saw on PBS, I would have never believed it. Now, tropical fieldwork is really amazing. I've been able to travel around the world collecting ants. I've been able to take students with me into the rainforest, which is really great, work with some amazing colleagues. This top right picture here is actually from French Guiana, where they have this really interesting system for accessing the canopy. A lot of the ants I work on are found only in the canopy. So what this is I look like that little spider-like object, and you have these lines where you can drive yourself over to trees with a little remote control and do your scientific research and then drive yourself up and over that tree to the next tree. It's really remarkable. I've had some of the best experiences of my life doing tropical field work. It also comes with its downfalls. Of course, I've been tremendously sick at times. And actually, this picture on the very bottom right is of a tick that I inadvertently smuggled home from Africa in my nose. It's now in a museum collection. So why is it so important to study biodiversity, especially in tropical rainforests? Of course, if any of you spent time there, you know that there are these amazing ecosystems with lots of diversity, and they can seamlessly go on for miles and miles and miles. But unfortunately, this is what's happening the world over. We're seeing conversion of that rainforest habitat by humans for use and for agricultural needs. So really, in order for us to be able to protect those regions, we need to understand what's there. So what I'll do is I'll walk you through some examples of the kinds of research I do on those specimens that I went out to the rainforest to collect, to talk about the work that I do firstly in evolution and biogeography. So biogeography is just trying to understand how species are distributed across the planet, right? Or wherever they may have their distributions. And in this case, we're also going to ask questions about their interactions with the flowering plants. So why would we expect feedbacks between ants and flowering plants? Unlike bees or other pollinators, where you can see the one-to-one relationship that as the flowering plants diversified and there were lots of new species, pollinators could take advantage of those resources, then the plants became more and more dependent on them for the pollination services. Well, ants don't actually engage in pollination. In fact, there's no known species that are good pollinators. So why would we expect that the ants rely on these flowering plants? Well, actually, one of my advisors, E.O. Wilson, and his colleague Bert Holdobler in 2005 had actually made a prediction. Because one of the things that we had known for a long time is that some groups of ants are found entirely only in the canopy. So they had predicted that those groups actually diversified or went through increases in speciation when the flowering plants came on the scene. But they didn't have the evolutionary tools to be able to address that. It was more of a speculation. In addition, one of the things we know about ants is that their diets have changed through time. The sort of ground plan earliest ants were predators. They lived in the soil and they hunted in the soil and on the ground. But as the flowering plants diversified, lots of other things did, and so it's speculated that they started to transition their food into broader and broader diets, including into plant-based diets. Now, from the plant point of view, one of the reasons we might expect that they would benefit from having ants around has to do with the fact that although ants do not pollinate plants, they are important for their biology because ants actually will disperse seeds. So the picture on the bottom shows an ant on top of a seed, and that sort of thing that looks like a toupee is a fatty body that was produced by the plant explicitly to attract ants to drag it away from the host plant. That thing's called an elaiosome, and the ant will eat it off and then discard the seed. And what this does is it increases the chances that the seed is dragged away from the mother plant, or the plant that gave rise to it, so there's not conflict between it and the parental plant. Now, this is thought to be so important to plants that it's evolved over 100 times across the flowering plants, and it's found in over 11,000 species. So 11,000 species of plants use ants to disperse their seeds. So in order to investigate these questions, one of the things we need to do is we need to infer a very large evolutionary tree. So this is essentially a phylogeny, and it tells us how species are related. But it doesn't just tell us how the species are related, and you don't need to see the tips, but what you need to see are the patterns that we can see in this branching diagram. Now, once we had that, we could actually start to ask additional questions. So the week that our paper was published, it actually got a lot of press. Every news source that I saw that covered our paper got it exactly right, except one, foxnews.com. And what they said was that we had analyzed the DNA of fossilized ants trapped in amber. Now, I wish that was true, some Jurassic Park-like technology where we go in with a big syringe and pull out a little ant DNA and then create a park of all giant ants. But it's not what we did, but we did actually incorporate the fossil record. It turns out ants have an incredibly diverse and very abundant fossil record. So once we had that phylogeny, we could go and look at fossils ourselves as well as look at the fossil literature, and we could use those to get an understanding about the age of the modern clades. So some of those fossils, although those species have long gone extinct, they belong to modern genera, and we feel very confident about that based on the morphological or anatomical characteristics. So once we went through, we were able to use 43 of those fossils to put a date on the age of ants. And interestingly, this is not common, both to have a large number of fossils, but they're also distributed across the entire phylogeny. It's not just in one little pocket. And those fossils range from as young as 15 million years old as 100 million years. So that allowed us to infer the age of ants. Ants are probably somewhere around 140 to 160 million years old. But the thing that really caught my eye when I first looked at this is that it looked like a lot of those branching events or those splitting events were happening in a very narrow window of the tree. But we actually had to test that statistically because one of the things we know about humans is we can discern patterns even when patterns aren't really there. So we use statistics to do that, and really what I want you to take away from this is just that the graph on the left shows us the present right here at 0 through 200 million years. So that's 200 million years ago. And we're looking at the number of the ant species as they increase through time, or we've binned them into their histogram or frequency. And what you see is right at 100 million years, there was a sharp increase in the number of ants on the planet. So we began to think about, well, what was happening on the planet around 100 million years ago? And it turns out that if you overlay on that phylogeny, that evolutionary tree, or that plot that I just showed you, where botanists say the rise and expansion of the flowering plant forest happened, we see almost complete congruence. But now this begs the question, were ants sort of just waiting for flowering plants to arrive and then following behind them as they expanded across the globe? So to begin to address that, we needed to think about where are ants found. And as I mentioned before, the highest amount of ant diversity is found in the tropical regions, right? And that as you move away from the equator towards the two poles, we decrease the number of species we see. So the warmer parts of this map tell us where there's more species. The cooler parts tell us where there's less species. Now, this pattern is called the latitudinal gradient in species richness. And what this means is that biologists have long observed that many groups of organisms have this same pattern, and the flowering plants are one of them. So maybe what's happening is the ants are just tracking right behind the plants. So to investigate this question, we had to infer an even larger evolutionary tree because we want to incorporate more information about where ants are found on the planet. Now, to try to understand this pattern we were seeing, we wanted to sort of contextualize that. So in 1974, Stebbins had come up with this hypothesis about why we see more species in lots of groups found in the tropics. And what he said is maybe it's that the tropics are acting as a cradle, where you have more species born at all the time, so you just end up with more diversity there. But on the flip side, he said maybe what we see is that the tropics are acting as a museum. It's where the oldest lineages hang out and persist, and the old lineages go extinct in other places because this is such a warm, stable habitat. So we set out to actually test this by using these expectations. So maybe on the one hand, what we see is that the tropics are acting as a cradle, where it's a species pump. It's just generating new species all the time. And if that's in fact true, what we should see is all the tips, or sort of the most recent parts of the tree, will be in the tropical regions, where maybe it's a museum, where we have sort of the species persisting through time. So all of those ancient lineages along the back part of the tree will be in the tropical regions. So in order to do that, we took every one of those species, and this is just a subset of them, and assigned them to the biogeographic regions. And so what you can see here is we split the world up into these 6 well-recognized biogeographic regions, and then we assigned each of those individual taxa on the tips or species to one or more of these biogeographic regions based on where they're found. So you can see this very first lineage is actually only found in 1 biogeographic region, where this one here is found in 4, and we did this for the entire data set. But if you remember, I told you ants are probably 140 to 160 million years old. And we know that these biogeographic regions were not always in their current configuration, right? So we know that as the tectonic plates sort of moved together, that we'd have to take this into account. So you can imagine that if we go back in time, so the sort of top panel, and you think about species that were found in South America, the probability that they could migrate to Africa more than 100 million years ago is 1 or 100%. Those continents were completely touching. Now, as they drifted apart through time, we had to account for that, so we decreased the chance that a species could migrate. In addition, we know some things like India, which was very far from Asia at 100 million years ago, the migration probability would be very low, but as it came closer and closer, the migration probability would increase. So we incorporated both that time aspect as well as their distributions. And here's what we found. So this is just a summary of what we found. And of course, what you'll notice right away is that most of the pies are reconstructed to green, suggesting that at least in these deep parts of the tree, the neotropical regions were critical to ant evolution. It's where we have the oldest lineages persisting through time. Now, I can't put the pie on all the tips because it would be very difficult to look at, but one of the things that if I did, you would see that green is incredibly abundant. In addition, if we take into account what we know about species there are more species in the neotropics and there are more endemic genera, meaning genera that are found in the neotropical region and nowhere else. Now, coming back to this question about ants just tracking behind the flowering plants, botanists have a pretty good idea when specific groups of flowering plants arrived in these biogeographic regions. So we went back and looked at that. And what we see is that the ants are not tracking the flowering plants. They aren't just following behind. In fact, what it appears they're doing is that ants were already widely distributed. They just were in very low abundance and low species numbers. And when the flowering plants came on the scene, they exploded and took advantage of this new habitat. And coming back to this hypothesis to try to understand why we see this disparity in where species are found across the planet, it looks like for ants, the neotropics have acted both as a museum where we have the deepest parts of the tree reconstructed, and also it's acting as a cradle where we see lots of new species arising. So if we come back to this idea about conservation and critical areas of the planet, at least for ants, the neotropical regions are a place we should heavily focus on to preserve sort of the deepest evolutionary history, but also ensure we have ongoing speciation. So thinking back to this about why ants would sort of diversify in the shadow of the flowering plants, I would argue that it's because prior to this, ants were living in the soil. And when the flowering plants came on the scene, the flowering plants did something unique. They created this entire leaf litter area where we know many species of ants live and feed entirely in that region, and they moved up into the canopy. So in the other kinds of forests, like conifer forests or cycad forests, we don't find ants at all living in the canopy. It's only in the flowering plant forests. So it allowed them to sort of expand both where they're foraging for food, but also, in fact, where they're living. So instead of having this sort of two-dimensional world, the world really became three-dimensional and allowed them to diversify. So next what I'll do is I'll take a moment just to talk about why I started studying bacteria. So as I mentioned before, one of the things we had seen is that ants were originally predatory, right? And then we have them expanding into the flowering plant forest, the canopy, and starting to take on plants into their diet. So thinking about how they would leverage that, very nutritionally poor resource and how that might also influence their evolutionary history, we started to investigate the role of bacteria in the guts of these ants and how it may have facilitated their shift into an herbivorous diet. In addition, not just into their diet, but even into the formation of their cuticle, and I'll talk a bit about that near the end. So why are gut microbes of ants so interesting? I would argue that they're a really great system to ask questions about, bacteria in these hosts because they have a lot of things that make them ideal to do these experiments. One of which, as I've already hinted at, they've evolved novel diets repeatedly. So if you want to understand the role of herbivory on structuring bacterial communities in the gut, we can have replicated experiments that evolution has played out for us. In addition, we can keep them in the laboratory and we can manipulate their diets and see how does changing your diets in real time affect the bacteria found in your guts. We also can study things like transmission, both on an evolutionary scale. We can ask sort of how did new groups of bacteria get introduced into lineages and do they persist through time? We can also ask the question, in a single colony, how are microbes shared among these individuals, right? We can do manipulations where we introduce pathogens and watch how they spread throughout these socially interacting organisms. In cases where these bacteria have been associated with their host for a long time, we might also ask the question, when do we see sort of shared evolutionary history? When do we see their evolutionary histories matching? And when do we not? And why is that? And lastly, because we know that ants are distributed across the globe, we can ask questions about where are they picking up these microbes? Is it sort of always from the same source? So do ants in the same environment always have the same bacteria? Or is it something about the food source they're eating in a particular region? So when I first started doing this work, I started it in collaboration with a long-term collaborator of mine, Jake Russell. And when we first started this project, I sort of felt like one of those old school rainforest explorers like Darwin or Wallace or Bates. Literally, like I was just running through the rainforest and grabbing everything I could see. And that's because we didn't know much at all about the bacteria associated with ants. So at first, all we were doing is just cataloging what we saw, right? Just documenting that diversity. And of course, we found a lot. So in our first study, we sampled almost 400 individual ants from 150 ant genera and just asked, what do you find? And maybe not surprising, there's a lot of bacteria there. But what really jumped out to us was this group of bacteria. It's in a group of bacteria in order called Rhizobiales. Now, some of you might be familiar with this group of bacteria because this is the same group of bacteria that are associated with the root nodules of leguminous plants, right? So in this case, these plants produce these specialized nodules on their roots just to house the bacteria. And in return, the bacteria fix atmospheric nitrogen for their plant hosts, increasing the nutrients available through the roots. So why are we seeing a group of bacteria that are related to these inside the guts of these ants? Now, another hint that made us think that these might be important to nutrition comes from the diet of ants. I would love to be able to go out and tell you definitively everything every species of ant eats. It's not possible. Of course, there are sort of some common knowledge in the literature that tells us sometimes this species has been observed killing this thing, but it's not enough to really give you confidence about what they're eating. So what we did is we used an analytical tool, which are called stable isotopes, to be able to put them on the food web. So along this bottom x-axis here, what we have things here that are herbivorous, right? So they're eating very low on the food chain. And at this end, we have species that are living very high on the food chain. Those are army ants right there. So we use this stable isotopic information to be able to place them with confidence on the food chain. Now, interestingly, if we plot when do we see ants that have association with this group of bacteria that I just told you is related to the one in plants, we only see an association with ants that are feeding low on the trophic scale. But if you have a keen eye, you may have noticed that there are 2 ants that feed very low on the trophic scale and have no association with this group of bacteria. And these are the carpenter ants and their relatives. And in this case, this is one of the few groups of ants that we know much about the bacteria associated with them. And in this case, the association has been so long and so conserved that the ant produces a specialized structure in its digestive tract just to house these bacteria. It's called a bacteriocyte. And in this case, we already know that these bacteria upregulate the host's diet, increasing the nutrient available to the ant. So it appears that if you're going to feed very low on the trophic scale, you have an association with this group of bacteria, Blockmannia and the carpenter ants, or with Rhizobiales in these other herbivorous species. Now, what you're looking at here is a phylogeny or an evolutionary tree of the bacteria that we sequenced from either the host or environment in which it came. And all of those in red are actually from ants. And you'll notice that almost all of those group together, suggesting that there's sort of one sort of association with ants, and then we have diversification of these bacteria. And interestingly, the names on the tips are of the ant host itself, and you'll see that ants that are related to each other have bacterial strains that are related to each other, suggesting that these bacteria have been associated with these ants for a long time. But interestingly, if we flip the question on the other side and say, okay, from the ant's point of view, what do we see? What we see is at least 5 independent associations of this group of bacteria with these ants. So it's not just that it's gotten in once and been tracking all ants, it got in each of these 5 independently and appears to be tracking these ants. In addition, we did some statistical analyses to understand the association of having these bacteria in your diet and where you're living. And we see a strong association with having these bacteria and being herbivorous, but also for living in the canopy, which we know is a place that does not have a lot of resources outside of plant-based resources, suggesting that if you're going to transition to being an herbivore, you're likely going to need to have an association with bacteria. But in the case of these bacteria, if they are associated with increasing the digestion of the host, we want to be able to show specificity of where they're found, right? They should be in the tissues where we know digestion's occurring. So in order to do this, we sampled ants, both their legs to account for what they just bump into in the environment. We sampled the mouth cavity so we could account for things that they might be ingesting, but aren't becoming part of their digestive tract. We then did careful dissections to look at 3 different parts of the digestive tract. We looked at the crop, the midgut, and the hindgut. And just to remind you, the crop is the social stomach. It's where an adult ant will go out in the environment, gather up some liquid resource, carry it back in the crop, and then regurgitate it to other members of the colony, both other workers and the queen, but also the larva. So it's the same thing that we see in birds, where they have a crop to store food that they come back and regurgitate to the young in their nests. Now, we know that no digestion begins in the crop because that's just a holding cavity so that they can carry food back. We know that digestion doesn't begin until it gets into the midgut or the hindgut. Now, in this case, we weren't looking at a single bacterium. We were looking at all the bacteria in these tissues. And this is just a subset of what we found, but I just wanted to demonstrate that right away, each of those tissues, so if you look down a column, we start seeing differentiation so that the crop is looking very different from the head and so on. But another way that we actually analyze these data is this way, with a PCOA plot. So what this is each of those dots is the entire bacterial community. And the more similar those bacterial communities are to another sample, the more likely they are to be in close proximity in space. So all of these samples that have this sort of broad distribution are from the leg, the mouth, and the crop. Now, once we move into the midgut and the hindgut, suddenly those bacterial communities, instead of being very diverse, are highly similar. And that's likely because the diversity is getting lower and the function is related to how they're upregulating the host nutrition. Well, since then, we've done a lot of work to be able to discern exactly the functional role of these bacteria in these turtle ants. And the first direct evidence we had is from doing metagenomic and genomic sequencing. And so this is work I've done in collaboration with Jake Russell, Yi Hu, John Sanders, and a whole suite of other people. And what we've been able to demonstrate is that in the bacterial genomes, they have all the genes to synthesize amino acids that they can then provide to the host, in this case, the ants. And in fact, that we see that these mechanisms within these bacteria are conserved for over 46 million years and are found across all the ants within this genus. Now, just because the bacteria have the capability to produce the amino acids doesn't mean that they're actually getting integrated into the host's tissue, right? This could be some mechanism just for the bacteria to exist in the gut. So we went in and did some analytical experiments where we provided antibiotics to some of the ants so that we could knock down or out their bacterial communities to control for the contribution of the bacteria. And then we fed them on careful diets to look at this. And what we found is only when we left the bacterial communities intact and fed them the stable diet do we see an increased enrichment of the essential amino acids. And these essential amino acids weren't just found in the gut. We were able to demonstrate that they're transferred across the gut lining into the hemolymph and are circulating within the host. So really suggesting that in this case, these gut bacteria are recycling urea from the host and then turning it into essential amino acids and then providing it back to the host. So if you think about having a coevolutionary history with a tight community in your gut providing some functional role, this is really remarkable that these ants are only able to live on this vegetarian or highly plant-derived diet because of the bacteria in their guts. But it turns out that the bacteria can contribute in other ways, and it's likely in the formation of the exoskeleton. So an exoskeleton for an insect is essentially its armor as it's walking around in the environment. It's a way of protecting itself from all of the predators and pathogens and even just physical objects it might bump into. Now, of course, this ant has a highly thick, tough, rigid cuticle, right? We can see that with our own eyes. But of course, there's a lot of variation. And ants have a lot of different kinds of armor. So one of the things that we know is that some ants have really rigid armor and some have very soft cuticles or exoskeletons. And interestingly, as I've been doing all of these dissections through my career, I started noticing something, that the ants with the thickest, toughest cuticles were either predatory or were incredibly herbivorous, and we're already starting to see that they always have associations with bacteria. So this led me to believe that there's something about a diet that's going to determine whether you can build a really tough cuticle. You're either getting the nitrogen from what you're eating yourself, or maybe you're relying on bacteria to provide that tough cuticle for you. So that's what we set out to test. Now, just to remind everyone, basic biology about an insect cuticle. Of course, it has distinct layers, right? So we know from our biology class that there are these distinct layers of the cuticle. But in addition, they're sort of built in these, three-dimensional structures to increase their thickness and toughness. And in addition, there's proteins that they're embedded in there as well. And another way that the cuticle can get tough is related to the tanning process. So before I told you that in those previous experiments, we were able to demonstrate that the bacteria could synthesize essential amino acids. And among them are the phenylalanine and tyrosine, which are the building blocks for the tanning and the melanization and sclerotization products. So remember, melanization is sort of what makes things dark, right? And we know that in some cases that darkness can impact toughness of materials, including cuticles. In addition, we can have the sclerotization process happen, but it all starts with these same building blocks. And so an insect cuticle is made of chitin, but in order for chitin to be attached to anything else, it requires a linker. Now, and usually what it wants to attach to, as I already alluded to, are proteins. But in order for a protein to attach to chitin, it relies on this imidazyl arm of histidine to be able to attach. And there can also be other embedded particles. One of the things we know is that many insects will embed melanin, as I've shown you, but also even metals. So some ant mandibles, there's metal that's embedded on the tips of the mandibles to reduce wear. So let's talk about this process of melanization. So what you're looking at here is a single mandible or jaw of a grasshopper, and you can see they've done sort of time slices to show you how it darkens through time. And if we look at what are the compounds found in that mandible, what we see is that carbon decreases through that tanning process, that oxygen stays the same, and it's an increase in nitrogen. So that's leading to that sort of toughness. And again, what we see is a sharp decrease in water, an increase in proteins. Chitin stays the same, but we also have these sclerotized tissues increasing. And so that's really what's leading to this toughness. So what we set out to do was to say, okay, we know that this group of bacteria, this group of ants has these bacteria that are synthesizing essential amino acids in the gut. Could that be contributing to the formation of the cuticle? Now, there has been a little bit of previous research on this, 4 studies in insects. And all of it's been done on beetles, and what they've been able to demonstrate is that when you give high amounts of antibiotics to these beetles, you see that their cuticle decreases in darkness. It becomes much thinner. In fact, you can see from that picture there, it's significantly thinner. And one of those studies even went so far as to show the metabolic pathway, the biosynthetic pathway, that leads to the formation of the cuticle. And what they showed is that the bacteria are responsible for all the precursor steps except the final step that the insect produces itself. So in this case, the bacteria are responsible for this increased hard or tough cuticle. So what we wanted to try to understand is which of these cuticular components might be impacted in the formation of cuticle in these ants. Could it be the protein? Could it be that linker, the chitin, or even the melanin that's seen in the beetles? And are there other pathways besides the one that's been investigated so far in this beetle group? And ultimately what we want to understand is, are these same pathways conserved? Are all the herbivorous and ants that have a thick cuticle using bacteria in the same way? So to do that, we took colonies of this ant, Cephalotes varians. It's my very favorite ant species in the world. It's one of the most beautiful ants in the world. It's the Florida Keys turtle ant. I'm sure you agree with me. And what we did is we split the colony in 2 and did some treatments. Half of them we left just as they were, and the other half that we gave antibiotics to knock down or out their gut communities. We then fed them a diet with labeled urea, which we labeled, we enriched the nitrogen 15 in this case. We then ran the experiment for 2 months, providing this new food diet almost every day. And the reason we had to do that is that we needed to account for individuals that had gone through the full developmental cycle and really incorporated both these, these compounds that are labeled urea, but also that had gone through the antibiotic treatments through every part of the development stage. So what we did is we caught a whole bunch of live ants, split them in two, and then we painted all of the workers that were alive when we started the experiment. So we knew who had not gone through this treatment. We then waited 2 months and waited for individuals to emerge, and then we were able to look at those individuals. We then looked at the gut tissue itself using some analytical chemistry techniques, as well as the cuticle itself, again using these analytical chemical techniques. And then again, we used scanning electron microscopy to see how the thickness of the cuticle was impacted. Now, when we did these analytical techniques, we used a method called NMR, and really all that lets us do is to trace a single isotope in this process. So in this case, we only traced with our liquid state NMR whether or not we had an increase in those essential amino acids that I talked about before. We did this for a couple of reasons. As I mentioned, these are sort of the starting compounds for that melanization and sclerotization process, so we needed to know whether it was impacting that process. In addition, it was a way of sort of double checking against ourselves, since we already knew from the previous work that bacteria were responsible for synthesizing these. And what we saw is that when we introduced antibiotics into their diets, we saw significant decreases in these essential amino acids being produced. And also as a check, one of the things we wanted to understand is, was everything affected when we did these manipulative experiments? And here what we see is that when we compare those ants that got the antibiotic treatment with those that did not, we see no difference. But in this case, what we're looking at is carbon-13, which we did not enrich, which is great. That's exactly what we should see. We should see no difference since we didn't enrich for this, suggesting that just by doing this manipulation, we're not changing all aspects of the chemistry within the host. But to understand what's actually happening, the first thing we had to do is we had to understand, what does just pure chitin look like in this analysis? So believe it or not, you can actually just order this from a molecular biology supply company. So you can get a giant jar full of just pure chitin. And when we analyze that, what we see is a peak that's quite distinct, and it lines up exactly where we think that the chemical shift should be for chitin. Now, if we take an ant that we did no experiments on and we have a lot of starting material for and do this same technique, what we see is a peak that's almost exactly aligned with the pure chitin standard, although much wider. And that makes sense because in this case, chitin we know is, of course, the exoskeleton is, of course, chitin, but also those embedded proteins, right? So that's what's leading to the sort of widening of this peak. Now, what happens if we go and look at our experiments? So these are the ants that we gave the antibiotic experiments to. Now, you might think right away, well, the peaks are much lower, and that probably is because of the antibiotics. But it turns out that's not why. It's that unlike when we could order a whole bunch of chitin from a chemical supply company, these were the ants that made it through the treatment all the way to the end, and then we dissected out just part of the exoskeleton. So the amount of starting material across even all the individuals we included was significantly lower. So if we sort of standardize that with the ants that we can get a lot of material from, you'll notice right away that we see that same chitin peak, right? And it's wide, including those proteins. But since these are the ones that we fed the enriched diet to, we actually see other peaks as well. So of course we see that chitin and protein peak, but here we see an enrichment in the peaks that are related to the guanine arm of arginine, suggesting that in this case, the ant is preferentially choosing some of these enriched bacteria to incorporate into this part of the biology. Now, if we overlay on this those ants that we did not give the antibiotics treatments to, so really these are the contribution from the bacteria, you'll notice a huge difference. Of course, we see a huge increase in the chitin and the proteins that are embedded within. We also see that in this case, one of the peaks of the guanine orange of histidine is higher from the contribution of bacteria, but both are still contributing, the host and the bacteria. Interestingly, we start to see an increase in the linker, right? Remember, that's the thing that lets chitin hold on to those proteins that are being contributed by the bacteria. And lastly, this is the thing that we found the most puzzling. What we see here is the remember that we needed the imidazole arm of histidine to attach to those proteins. We see an enrichment here, but what you may have noticed is in all other cases when I showed you these chemical structures, all of the ends are colored pink because those are the nitrogens that were enriched in our experiment. But in this case, only one of the nitrogens in that aromatic ring is enriched. And this was quite puzzling because we didn't quite know how to explain it. And we weren't certain that maybe we were missing something that we should have seen. But lucky for us, in the 1980s, Schaefer and colleagues had published a paper where they were doing some experiments with insects and looking at similar questions, but not related to the contribution of bacteria, just how is the cuticle synthesized. And what they were able to show is, again, that they found a peak that corresponds to the cuticle and the proteins that are embedded in it. But in relation to the imidazole arm of histidine, they found 2 peaks where we only find 1. So we know definitively that we're not seeing an enrichment in that particular nitrogen within this compound. We still don't know how entirely to explain it, and it's ongoing research. But coming back to this, using this scanning microscopy to look at how the cuticle is impacted, these figures are not to scale, so you have to look at the numbers, but you'll see that when we introduced bacteria, we saw a sharp decrease by about half in the thickness of the cuticle of these ants. But coming back to what had been observed in beetles, we actually saw no change in the color. So the melanization pathway does not seem to be impacted at all from these bacteria or the antibiotics. So what we've been able to demonstrate through this experiment is that we know that the bacteria are synthesizing essential amino acids that the ants are using. In this case, it does look like they're in fact incorporating them into the exoskeleton. And using the analytical techniques we used, we've been able to demonstrate that the cuticle, there's a contribution in the formation of the cuticle related to the protein, the linker, and the chitin, but not in the melanization. And we actually have now demonstrated a different biosynthetic pathway that explains the formation of the chitin. So it's really interesting thing to think about how these bacteria are providing resources not just to the upregulation of the diet and sort of nutrition overall, but into a basic part of the biology of this organism in building the cuticle. But being an evolutionary biologist, of course, I love drilling down to the actual function of these interactions and the mechanisms that are involved. But really, I like to then take a step back and say, okay, well, now we know something about these bacterial communities associated with ants. So we could probably make predictions about how this will play out. So I've been talking a lot about this group of ants, the turtle ants. We know that they have this very long evolutionary history, about 46 million years, with bacteria in their guts. Each of these pies represent a dissected gut and all the bacteria in it. And what you see is if we compare that ant to a distantly related another herbivore, their bacterial communities are more similar, even though distantly related, than to even other species within the same genus that have an omnivorous diet. So when I first started this project, I told my students, okay, well, as a comparative biologist, we need to make, we need to compare herbivores, generalists or omnivores, and predators. And what we're going to find in the predators is really simple bacterial communities because their diet has everything they need. That you should never say that out loud to your students because it's a guarantee you will be wrong. And of course I was. So I was incredibly shocked when these data came back. Not only are they really diverse, those bottom 3 pies are ants from the same colony. There seems to be no overlap in their bacterial communities at all. And when I was thinking about this, I actually realized that I had made a few mistakes in designing this project. One of which I said, all right, we need a predator. And this is a bullet ant. And bullet ants will take down live prey. But they'll actually also eat plant-derived resources. If they find something like an extra flow of nectar, they'll sit there and drink that for hours. They love sugar water. So I thought, okay, well, they do incorporate plants into their diets. The other mistake I made was thinking about the biology. We tend to think of predators as going off on these giant foraging raids and taking down live prey and then dragging it back to the nest. Well, these are not sort of these group hunters. They're actually solitary hunters. Every ant leaves a nest and goes in a different direction. So I thought about it again, and I thought, okay, well, I suspect that a lot of this is just contamination based on what they ate last. So the way I like to think about it is if we would have sampled all of our gut communities this morning, those of you that live in the same house or happen to eat breakfast together, your communities might look similar, but pretty much overall we'd all look different. Now, if all of us went out to dinner tonight together and we sampled our gut communities again, suddenly they look highly similar. And that's because we know 2 things. One, there is contamination in all of the food that we eat. And if we just do a quick snapshot, we will pick up some of that contamination. In addition, the bacteria that are present in our gut and are important to our digestion, we know that they respond to different foods. So if you eat a high sugar diet for a very long time, some bacteria go into sort of higher numbers. If you eat a lot of meat in your diet, other bacteria will grow into large numbers. So I thought, okay, I need to redesign this experiment. So I took my entire lab down to Costa Rica, and we collected lots of colonies again of these bullet ants. But this time, we captured some of those individuals, those wild type individuals, right? So what they looked like in nature. And then we brought lots of them back to the lab and fed them on specialized diets that we were controlling that were microbe-free to see what happens to their bacterial communities. So the way I'm going to present these data is a bit different. It's through what's called a network-based analysis. It's not complicated. In fact, it's quite intuitive, so I'll walk you through it. Every one of those dots is one ant gut on all the bacteria that we found in it. If it's colored red, it was those wild-caught individuals that we caught right in nature. If it's yellow, it's the individuals that we fed on a sterile diet, but from the same nests. Now, each of those lines tells us about whether a bacterial is unique to that sample, or more importantly, if those balls are close together in space and there's a lot of connections, it tells us that the bacteria in those two samples are highly similar. So I sort of think of them as magnets. So when you flip them one way, they're attracted to each other. When you flip them another way, they're repelled. So if the bacterial communities are very different, they're very distant from one another. And here's what we found. So those red samples are those wild-caught diets, and that mirrors almost exactly what we saw before. A lot of bacteria there and not a lot of similarity. Once we fed them on that controlled diet, what we saw is that diversity decreased and the similarity between all the samples became highly similar. And keep in mind that these red dots, some of those are the sisters, right, from the same colony of those yellow ones, suggesting that what we see is a lot of contamination that's just passing through the digestive tract and is not important in these species. So what I hope I've been able to convince you is that by studying biodiversity and ants in general, we can ask some really fundamental questions in biology. What I've showed you today is that ants are an old group that likely diversified in response to the expansion of the flowering plant forests, that the tropics and the neotropics in particular were really critical to the evolutionary history of ants and the ongoing speciation within the group. That thinking about the role of symbiotic bacteria, even though I'm an ant biologist, really has an important role because it's impacting interactions of those animals with the world around them. And in this case, it's facilitated them to be able to shift on into an herbivorous diet and expand into this novel niche called the canopy. And even in this case, not only are they synthesizing essential amino acids, we also know that they're even important in things like the formation of the cuticle. So I've been really fortunate to sort of been able to take advantage of the diversity of ants and all the research questions we can have and travel around the world collecting them, seeing nature at its finest. And I would actually argue that being outside is actually what, in many cases, informed the questions that I ask. I never thought that I would start studying bacteria. It was only when I started thinking about how do ants take advantage of these diets that they didn't start on in their evolutionary history. Or why is it when I'm dissecting these ants that I want to understand something about their diet? Is their cuticle so different, right? So I think that spending time outside and knowing your organisms well has really benefited my research program. So of course, there's many people to thank. I always have a lot of collaborators. I've been fortunate to have some amazing students in my lab, some of which I've seen in the room today. And of course, my funding sources. And if there's time, I'd be happy to take questions. Thank you so much.